Alright, hello and welcome back to part B of this very entertaining video. We've kind of laid out the histology, we've seen the anatomy, we kind of understand how and where things are. Now what we'd like to do is focus on the specifics of how things interact with each other, specifically how to initiate digestion, how to turn it on, and also with negative feedback, how to turn it off. And to do that, we'd like to take a closer look at how all of these cells, the chief cell, the parietal cell, and my neuroendocrine cells, are working together to regulate this system. So let's reintroduce these cells. We'll start first with the chief cell. And if you recall from the previous video, the job of the chief cell was to secrete pepsinogen. The next cell is the parietal cell. We ascribe two different functions to the parietal cell, the secretion of hydrochloric acid and also the secretion of intrinsic factor. All right, the next cell we want to discuss is the G cell. And for reasons that we'll discuss here in just a second, we're going to draw the G cell with a belt around its waist. Nice belt buckle. And we said its job was to release gastrin. All right, next up is the ECL, which releases histamine. And lastly, the D cell, or delta cell, more appropriately. We said this was the off switch, as it releases somatostatin. All right, now let's see how all these things are working together. The G cell, we said, was the on switch. There are a couple of ways to turn it on. For example, it's got a receptor. This is going to be reaching out into the lumen of the stomach, a receptor for amino acids. However, the biggest factor in turning the G cell on is stomach distension. And that's why we drew it with this belt. As the belt stretches, in other words, when I put food into my stomach, these cells stretch. And it's like popping the snap off of your pants after eating a big meal, right? As this cell stretches, it releases gastrin through exocytosis into the blood. Now, there's a couple of things that I want you to notice in my drawing. The first two cells on the left, the chief cell and the parietal cell, they're showing exocytosis on the top half, in other words, into the lumen of the stomach. The other three cells, my neuroendocrine cells, the G cell, the ECL, the D cell, they're exocytosing cells through the basolateral surface getting into the blood. And that's the idea of endocrine signaling, right? Where hormones get into the blood, and it's through the blood that they communicate their effects. Now, let's follow gastrin and see what it does. As this is the on switch, let's first take gastrin to the chief cell. So here's a receptor on the chief cell. Gastrin stimulates the chief cell. Other stimulations for the chief cell include acetylcholine, if you recall, as the neurotransmitter from the parasympathetic system. So this is going to be postganglionic parasympathetics releasing acetylcholine onto muscarinic receptors. As we trigger the chief cell, we're going to drive exocytosis, and we see the release of pepsinogen. Now, pepsinogen is going to be an important part of starting the process of breaking down proteins. But as pepsinogen, it still is inert. It can't do anything. It needs an activation signal. So let's also look at the effects of gastrin on the parietal cell. Stimulation of the parietal cell by gastrin primarily drives the production of hydrochloric acid. And it does this by activating a primary active transporter. So we're using ATP to pump out protons in exchange for potassium. We saw these same transporters in the kidney, where protons and potassium were exchanged as a regulatory mechanism. One positive cation for another. Of course, as we think about our buffer system, which hopefully is ingrained in our heads by now, we've got carbon dioxide in equilibrium with H plus and bicarb. And as the stress is to decrease H plus as it's pumped out of the cell by primary active transport, this, of course, drives this reaction toward the production of bicarb. As bicarb levels go up, I hope you're seeing common themes here, the bicarb gradient is going to drive the antiport exchange for chloride, bringing chloride into the cell and increasing the concentrations of chloride within the cell. Of course, now we have a leak channel on the luminal surface for chloride. Chloride goes out into the lumen, and what do we have? We've just made hydrochloric acid. The chloride here, and the H plus here. There's our hydrochloric acid. Now we can come back to pepsinogen because this is the signal to activate pepsinogen. At low pH, pepsinogen is activated by this acid to an active enzyme called pepsin. And as we'll detail later on when we focus on amino acids, pepsin is all about breaking down protein. Now, we did also state that in addition to acid, the parietal cell is releasing intrinsic factor, and this is important for vitamin B12. Without intrinsic factor, vitamin B12 cannot be absorbed actively in the small intestine And in the absence of vitamin B12, this is going to lead to problems, specifically certain types of anemia, as vitamin B12 is important in red blood cell formation. Now, there are a couple other triggers on the parietal cell that we want to be aware of. Again, we see the presence of receptors for acetylcholine, emphasizing the importance of the parasympathetic system in activating this. We also see a receptor for histamine. And that leads us to our next cell, which is the enterochromaffin-like cell, the ECL. Not surprisingly, it's gastrin as well that's driving this, among other things. And the release of histamine specifically functions to stimulate acid production from the parietal cell. And I want you to appreciate why that is, right? What's the primary function of acid? Now, we see acid here taking pepsinogen and converting into pepsin. I get that that's important, but it's not the primary reason we produce acid. The primary reason for acid production is all about sterility. Think of all the stuff that you're eating. You've constantly got your hands in your mouth, or you're eating this hamburger. Of course, your hands are all over the hamburger. There are a lot of bacteria, and I'm not saying it's a bad thing, but there are a lot of bacteria that get into your stomach. And this is the body's primary mechanism for killing off a significant portion of that bacteria, sterilizing the environment to protect what's coming into the body. And so it makes sense for histamine, which is associated with the innate immune response, to be able to trigger acid production independent of food intake. Of course, we can't forget to emphasize the fact that really all of this is turned on also by acetylcholine. The parasympathetic system is a powerful driver of all of these systems. I can activate this, in other words, simply by thinking about eating food. And you can feel the saliva build up in your mouth. That same sort of glandular activation is happening in the stomach in preparation for the food that I'm about to eat. All right, let's look at the delta cell now. We said this was the off switch, and not surprising, it's also stimulated by gastrin. We can see how gastrin stimulates all of these cells. But now we're talking about negative feedback that's going to cause the system to shut down. So with the exocytosis of somatostatin, somatostatin really has two primary targets in this. It's going to come back and inhibit the release of gastrin from the G cell, and it's also going to inhibit the parietal cell to make sure that I'm stopping acid formation. And so generally speaking, when we think about somatostatin, you can think of this as a pan inhibitor. In other words, an inhibitor of a lot of different things. Now, maybe it's worth mentioning there are other enteric neurons, right? The NANC neurons, non-adrenergic, non-cholinergic. There are other stimulatory, inhibitory mechanisms at play here, but this provides the best picture for how all of these cells are working together. Starting again with the G cell, this is my on signal, triggered primarily by stomach distension. In other words, there's something in the stomach, there's food in the stomach, causing the stomach to stretch. That's going to lead to the release of gastrin. Gastrin turns on the chief cell, which causes the release of pepsinogen, which will be converted to pepsin by acid produced by the parietal cell. Pepsin is an active enzyme involved in the digestion of protein. The parietal cell uses gastrin as a trigger to run a primary active transporter that exchanges protons out into the lumen for potassium. This is one of the body's key ways actually of absorbing potassium and cannot be discounted. Regardless, here we're pumping protons out and we're using that to create a bicarb gradient that helps pull chloride in, and then chloride can move out into the lumen as well to create the production of hydrochloric acid. We also mentioned intrinsic factor and its role in vitamin B12. On the other side, we've got the ECL releasing histamine as a means to vamp up acid production because acid is the key sterilizer for the stomach. And then to round it all out, we've got the delta cell releasing somatostatin in a negative feedback loop to make sure the system gets turned off at the appropriate time.